Cells are not quiet systems. In their interior, proteins consume energy, generate forces, and reorganize structures without rest. Understanding how to measure that distance from balance is one of the great challenges of today's biophysics. If you like this content, do not forget to subscribe and give it like. Your support is fundamental to continue sharing interesting information about biology and science. The work of Narinder and Fisher-Friedrich, published in Physical Review X, goes into the behavior of 3 different measures of imbalance in living human cells. The study focuses on the cortex of actin, a network of proteins located just under the cell membrane. The cortex of actin is a network of protein filaments that gives shape and resistance to the cell. This network converts the cortex into an active system that produces movement thanks to a continuous supply of energy, being associated with motor proteins such as myosin. To study fluctuations in the actin cortex, the authors used atomic force microscopy, AFM. They recorded how the position of a tiny tip inserted into the human cell cortex in mitosis, acting as a mechanical sensor of internal activity, fluctuated. Measurements showed that the tip not only vibrated softly, but also experienced abrupt peaks. These events revealed a rapid rise and slower relaxation, indicating the presence of active processes beyond simple thermal noise. In equilibrium systems, there is a relationship between fluctuations and mechanical response, known as fluctuation-dissipation theorem. When this relationship is violated, the system is out of balance, and an effective temperature can be defined to quantify that violation. The researchers found that the cell cortex does not behave as a balanced system. Fluctuations in long timescales were much more intense than expected, indicating that internal activity generates additional energy. In addition to the actual temperature, entropy production is another relevant measure. By inhibiting the activity of myosin, they found that the production of entropia decreased, although the effective temperature did not always follow the same trend. Another aspect of the study focuses on temporal irreversibility, analyzing whether trajectories are the same when reversing the sense of time. The authors found that the measure of irreversible decreased by reducing motor activity or actin replacement. The study concludes that effective temperature, entropy production, and temporal irreversibility are not interchangeable. Each one captures a different aspect of behavior outside of balance, which is crucial to understanding living systems.